Today we are going to talk about neuromuscular junction, how the information transfer from the neurons to the muscles, right? That is also called neuromuscular transmission. You know that information comes from to the skeletal muscle. Let's suppose I'm talking about my biceps muscle. It is connected with the spinal cord with the motor neurons. So the motor neurons in the spinal cord which are supplying the information to the biceps skeletal muscle, and we have to see how this information moves from the motor neuron to the skeletal muscle membrane, right? Let's suppose that here is your spinal cord, right? And this is a section of spinal cord, and you must be knowing that there are anterior horn and there are posterior horn. And anterior horn are motor horn, from where the motor neurons come out. These motor neurons which are coming out from the anterior horn, they are destined to supply the neuromuscular junction, the junction between the nerve and the muscle, right? So what really happens, this is a neuron which is coming out, and this neuron, let's suppose, is, this is a neuron here. This is cell body, right? This is the axon coming. I have enlarged the ending of this neuron, and ending of this neuron is called nerve terminal. And this nerve terminal is connected with the muscle. Is that right? Which is innervating the muscle cell and stimulating the muscle cell, which is response. Let's suppose I draw a muscle cell here. This is a muscle cell, and where muscle cell become near to the nerve ending, there it makes multiple folds, and this is, suppose here is a muscle cell. Now what we have to see is, what we have to learn today, that how action potential coming on the motor neurons, right, bringing electrochemical signals or electrical signals. Action potentials are electrical signals. When action potentials or electrical signals which are coming from the spinal cord on the membrane of the motor neuron, when they reach at the nerve ending, right, how they take the information, stimulatory information from the nerve ending to the muscle. Is that right? Actually what really happens, that action potentials which are coming on the motor neuron surface at the nerve ending, they lead to the release of chemical neurotransmitter, and that chemical neurotransmitter will bind with the receptors on muscle membrane, right? This part of the muscle membrane, this part of the muscle cell membrane is called motor end plate. What is it called? Motor end plate, because this part of the membrane of the muscle cell is connected with the motor neuron, and this plate of the membrane, it will receive the chemical message from the nerve ending, and then this plate will convert the chemical message into electrical message. I will explain everything step by step. First of all, information coming as electrical signals in the membrane, right? From here chemical substance come out. It means electrical signals are converted into chemical signals. Then chemical signals, the chemical substances stimulate the nerve motor end plate, and then at motor end plate again electrical signals are generated. Is that right? And of course later on we'll study those electrical signals which are generated here, their action potential in the what? Muscle membrane, which will eventually lead to the stimulation of the muscle contractility machinery, which we'll discuss later. Today's topic is only to understand how the action potential or electrical information coming on the neuron surface is converted into chemical information or chemical signals, and how chemical signals go to the to nerve ending, motor end plate, and when chemical signals come to the motor end plate, how they again convert the chemical signals into electrical signals, right? And this transfer of information or signals from the motor nerve ending to the motor end plate, this whole information process which is transferred, electrical information, chemical information, again electrical information, this process is called neuromuscular transmission. What is this process called? Neuromuscular transmission. That there is transmission of signals from the neuron to the muscle. So this is about neuromuscular transmission. Is that right? And this area where nerve ending, right, motor nerve ending and motor end plate from the muscle, they are coming together. Whole this area is called neuromuscular junction. What is it called? Neuromuscular junction. So what we say that at neuromuscular junctions there is neuromuscular transmission. This is our today's topic. What is neuromuscular junction and how neuromuscular transmission occurs. Simply what we are going to discuss, how the motor neurons talk to the muscle. How the motor neurons talk to the muscles, that muscles should contract. That's what is our today's work. Okay. First of all, as I told you, when electrical signals are coming here, they lead to the release of chemical substances. So it means this nerve ending should have some chemical substances which should be released as chemical signals. Those chemical substances which are accumulated and stored in this nerve ending, those chemical substances are called neurotransmitters. What are these? Neurotransmitters. What are neurotransmitters? These are special chemical substances which are present in the nerve endings, and they are released from one nerve ending, right, to transfer the signals to the postsynaptic membrane. The junction of the, look, what is synapse? Synapse is the meeting point of the membranes of two cells where information can be transferred from one cell to the next cell. What is synapse? Synapse is a meeting point of the two membranes of two cells where information from one cell can be transferred to the other cell. And because at neuromuscular junction the synapse is made between the neuronal membrane and muscular membrane, and in this synapse the information passes through a chemical neurotransmitter. That is why neuromuscular junctions are classified as chemical synapses. How they are classified? They are called chemical synapses, because in this synapse, in this contact point, right, information is transferred from one cell to the next cell by the chemical substance. So we call these such type of synapses chemical synapses. So when we study in detail neuromuscular junction and neuromuscular transmission, we are studying a classical case of chemical synaptic activity. Am I right? And this membrane is called presynaptic membrane, and this is called postsynaptic membrane. Presynaptic membrane will talk and postsynaptic membrane will listen. Presynaptic membrane will release the chemical substances and postsynaptic membrane will have a receptor for that. It keeps on talking, it keeps on listening. This is something like ideal couple as female imagine. What is the ideal couple? Female always talk, men always listen. Is that right? So it is only one-way transmission. This is only one-way transmission, right? You cannot get the reverse direction. Is that right? The neurons always talk and muscles always listen. Is that right? At neuromuscular junctions. At neuromuscular junction. So this transmission is two-way or one-way? One way. It is one way. So let's see how the neurons talk and how the muscles listen. And after that, how the muscles start their action. Right? That we'll discuss later. Okay. First of all, they should have a chemical substance. So from the chemical substance at neuromuscular junction at the skeletal muscle is acetylcholine. The chemical substance is acetylcholine. So within these neurons, there are special type of membrane-bound vesicles, and in those membrane-bound vesicles, these are the molecules of, yes, acetylcholine. There are thousands and thousands of the molecules of acetylcholine well concentrated in the synaptic vesicles of nerve ending. Is that right? Nerve ending has thousands of such membrane-bound vesicles, and they are loaded with acetylcholine. it means that we should know from where the acetylcholine comes and from where the vesicles come. Is that right? We should know from where the acetylcholine comes and from where the vesicles come. First of all, we talk about these vesicles, right? These vesicles, from where they come. Yes. Who has any idea from where these vesicles come, which are loaded with acetylcholine? Right. Are these vesicles synthesized in this nerve ending, or are these vesicles coming from the cell body of the neuron? Yeah. The question is that that there are thousands and thousands of vesicular, you can say vesicles there, in which you can store the neurotransmitters. These bags of the neurotransmitters are basically membrane bound vesicles with very special type of proteins which help in storage and capture of the, you can say, neurotransmitter. These vesicles are not synthesized in nerve ending. These are synthesized in the cell body. Why they are synthesized in cell body? Because these vesicles have very special type of proteins in their membrane. And you know, proteins are synthesized from messenger RNA. You know, messenger RNA are translated in protein, and messenger RNA are produced by the DNA transcription. So it means DNA should be transcribed into messenger RNA, and then messenger RNA should be translated into proteins. And if this vesicle has proteins, then those proteins must be coming from some place where there should be DNA. And there's no DNA in this nerve ending. Where is DNA? Cell body. So that is the logical reason that why actually all the different type of proteins, listen carefully, all the different type of proteins which are present on this very specialized nerve ending, all those proteins are synthesized in the cell body and transported all the way down to this nerve ending, because nerve ending does not have nucleus, nerve ending does not have genetic material, it does not have genetic program to make proteins. So this is the basic principle in all the neurons. Whatever proteins are present at nerve endings, there are proteins which help in storage of neurotransmitters, there are proteins which help in movement of the vesicles, there are proteins and enzymes which help in synthesis of neurotransmitters, there are proteins which help in the uptake of neurotransmitter or reuptake of neurotransmitter or uptake of neurotransmitter precursor molecule. All these proteins which are present in this nerve ending, they are coming from, yes please, cell bodies. And then gradually, very, very slowly, through the exoplasm, they are coming to the nerve. Out that these vesicles, which are having special type of, let me draw one vesicle out. Let us suppose this is vesicular membrane. Actually, this membrane has very special type of transporter proteins within it, right, which can transport that neurotransmitter and concentrate it inside. Number one. Number two, these vesicles have also another special type of proteins which help the vesicle to fuse with the neuronal membrane. So these vesicles are loaded with special type of protein. Then, as I said, that proteins can only be synthesized in the cell body and not in the nerve ending. So these vesicles are synthesized in the cell body, right, where we have the DNA, we have the genes which will be transcribed into messenger RNAs, and those messenger RNAs will be translated into different proteins, and all those proteins will come down to the nerve ending through the axon. Here, some proteins will be part of the vesicle, right, and pumping the neurotransmitter in. There will be other proteins on the surface of vesicle which will help the vesicle on stimulation to bind with the neuronal membrane, right. But when these vesicles are produced and they are coming down through the exoplasmic flow and they reach here, originally these vesicles are empty. These are empty bags. These are empty bags. Let's suppose this is a bag which is empty bag and it doesn't have any neurotransmitter. Now, from where the acetylcholine comes? Acetylcholine is, okay, I will ask you, acetylcholine is produced in the nerve ending or it is coming from the cell body? Answer is absolutely wrong. That acetylcholine is not coming from the cell body. It is a very small molecular weight neurotransmitter all the small molecular weight neurotransmitters are synthesized in the nerve terminal. Is that right? So it means actually acetylcholine is synthesized in the nerve ending, but the enzymes and different proteins which are required for synthesis, storage and release of the acetylcholine, all those proteins are coming from the cell bodies. So you can say the machines, right, the factories which synthesize, store and release the neurotransmitter, those proteins are coming from the cell body, but acetylcholine is synthesized locally in the nerve ending. So nerve ending is semi-autonomous unit which can synthesize the neurotransmitter, which can store the neurotransmitter, which can release the neurotransmitter, and sometimes, not in case of acetylcholine, but sometimes some neurotransmitters by some nerve endings are recaptured. So nerve ending is a very, very important biological semi-autonomous functional unit. So we see how this particular specific type of nerve ending works. Actually, this nerve ending has proteins in its membrane, and these proteins are choline transporters. These proteins are choline transporters, and what these proteins are doing, they are actively transporting the choline from extracellular fluid to the nerve ending. In this way, from where the choline is coming, of course you must be knowing you have a gastrointestinal system and you eat so many funny things, and some of these things have choline, and choline is absorbed from the GIT into your circulatory system, and then choline comes to extracellular fluid all over the body. So all over the body extracellular fluid is having choline, and wherever these such nerve endings which are cholinergic nerve endings are dangling into what? Extracellular fluid, they have these special transporter which are helping these nerve endings to concentrate the choline from extracellular environment into intracellular area. So they are choline transporters in the nerve ending membrane, and of course you will remember these choline transporters are coming from the cell body because they are special type of proteins. Is that right? But don't tell anyone that choline is coming from all the way down, right? So once choline come out, now actually to make acetylcholine, we should fuse the choline with acetyl-CoA, right? Acetate, with acetate. So what really happens that there are mitochondria in the nerve ending. There are mitochondria in the nerve ending, and mitochondria provide acetate. You know, mitochondria synthesize, mitochondria have acetyl-CoA, and acetyl-CoA can be converted into acetate or simply acetyl coenzyme A. Now acetyl-CoA is coming from the mitochondrion, is that right, which are present normally in the nerve ending, and choline is captured by the special transporter from extracellular environment and concentrated into this. Then this acetyl-CoA and choline, they are put together and they are fused, and when they are fused together, right, this choline and so acetyl-CoA and choline, right, they are fused together, and when they fuse together they make a molecule, and that molecule is called acetylcholine. Now this acetylcholine formation, right, is done by the help of an enzyme, and what is the name of that enzyme? Choline acetyl transferase, right? The enzyme which is doing this function, that enzyme is called, this is the enzyme, and this enzyme is responsible for this reaction. Is that right? And that this enzyme is actually helping the choline and acetyl-CoA to come together and to make acetylcholine. So what is the name of this enzyme? Yes? Okay, little bit. What was this transporter? Choline transporter. And this enzyme, right, it is called choline acetyl transferase. Now, from where this enzyme is coming, right? From where this enzyme is coming? Can you tell me? This must be coming from the cell body because enzyme is a protein. It's a difficult thing. It's a basic principle, isn't it? All proteins are synthesized from the cell body and transported to this very special small autonomous factory of neurotransmitter synthesis, storage, and release system. Is that right? So up to now, what did we learn? That nerve ending is having choline concentrating transporters, and they have mitochondria which provide acetyl-CoA. So the choline and acetyl-CoA are fused together by the help of choline-acetyl transferase, and the resulting molecule is called acetylcholine. And this acetylcholine should be appropriately stored. And for storage purpose, what really happens that your friend, I told you there are vesicles, right? And these vesicles are having very special type of, what are these? Transporters. What are these transporting proteins? These proteins are actually sensitive, transporting proteins are sensitive to the presence of acetylcholine, and as soon as acetylcholine is formed, it is transported into the vesicle, and then your vesicle become very rich in acetylcholine. So acetylcholine is concentrated into these vesicles. Am I clear? No problem up to this? In this way, we have synthesized the chemical signaling molecule and stored into the nerve ending. Now we have to see still another step, that when electrical information or wave of depolarization is coming here, how it leads to the release of neurotransmitter, and how neurotransmitter binds with the post-synaptic membrane or with the muscle end plate, and how on the muscle end plate electrical events start, and eventually muscle membrane develop action potentials. We have to discuss how the action potential coming on the neurons convert into chemical signals and chemical substance is released, and how chemical substance generates local potentials and eventually action potential on the membrane. Now let's start with it. All of you must be knowing that when action potential is coming here on the membrane, it means wave of depolarization followed by the wave of repolarization is approaching the nerve ending, right? So when spinal cord stimulates this motor neuron, wave of depolarization is moving down. You know, when wave of depolarization is moving down, what are these channels? Voltage gated sodium channels. So voltage gated sodium channels bring lot of sodium N, and this when wave of sodium influx is coming, naturally what happened that if resting membrane potential was minus 70 millivolt, so as soon as sodium comes in, right, then membrane become depolarized, maybe it become plus 10 millivolt. Is that right? So what is really happening? That when wave of depolarization approaches to the nerve ending, it produces heavy sodium influx, and that sodium influx reverses the polarity of the inner side of the membrane from resting membrane position to depolarization. So as soon as nerve ending is depolarized, now this is the point to understand, as soon as action potential or wave of depolarization approaches the nerve ending, and as soon as nerve ending is depolarized, nerve ending activates its very special depolarization sensitive calcium channels. What are there? There are depolarization sensitive calcium channels. So these depolarization sensitive calcium channels, they will be activated, and you know calcium is well concentrated inside the neuron or outside the neuron. Yeah, calcium is more concentrated in extracellular fluid. So as soon as you can say wave of depolarization reaches here, as soon as wave of depolarization reaches here to the nerve ending, it will produce the depolarization of the nerve ending, and then depolarization sensitive, for example, it's become plus 10 millivolt, right? And you can say that nerve ending is depolarized, then depolarization sensitive calcium channels will open, and there will be calcium influx. Am I right? There is calcium influx. right? So you say one electrical information is converted to another. The originally action potential came as a wave of sodium influx, and that triggered in the nerve ending wave of calcium influx. When calcium comes in, then what happens? When calcium comes in, actually there are calcium sensitive proteins which are present on the surface of the vesicle, right? The special type of calcium sensitive proteins present on the surface of the vesicles, which are normally present all the time on these vesicles, and the calcium sensitive proteins also present on nerve cell membrane. So these are special calcium sensitive proteins. Now look, actually if this protein dock into this, if this protein fit into this pocket, both proteins will interact and contract, and this vesicular membrane will fuse with neuronal nerve ending membrane. Is that right? But these proteins can be activated by calcium. What really happens, as soon as calcium binds at these ends, some calcium at this point, some calcium at this point, both of them are positively charged, so they will be repelled. So what really happens, as soon as calcium comes in, this open up because calcium is binding here. And calcium brings some changes in these proteins also, and they are more ready to bind here. So what is the real result? That in the presence of the calcium, these proteins which are in the membrane, right, they come into active configuration, and the proteins of the vesicular membrane, they also are able to interact here. When these two proteins interact with each other, right, I've shown of course these proteins as very big molecule, but in proportion these proteins are extremely small molecule on vesicular membrane as well as on nerve ending membrane. So when these proteins interact, they help the both membranes to fuse with each other, and when both membranes fuse with each other, what really happens, situation become like this, because the point where vesicular membrane and nerve ending membrane fuse, membranes dissolve, and a gap is generated through which your beautiful stars will come out. What are these? Acetylcholine. In this way, there is release of neurotransmitter from the nerve ending. This process is called exocytosis of endocytosis means taking up some material. Exocytosis means release of some material. This is also called exocytosis of neurotransmitters from the nerve ending, which is calcium influx dependent. Let me tell you the name of these proteins, right? These were the proteins. This was one protein which was present on synaptic vesicle, and there were other protein which was present on the membrane, right? These proteins. This protein is called synaptobrevin. And there are many other proteins also like this. And this protein is called syntaxin. And there are many friends of syntaxin also, right? So these proteins actually are active. Their fusion is activated by calcium influx, and these proteins help the vesicular membrane to fuse with the nerve ending membrane, so that both membranes fuse with each other, produce a gap through which neurotransmitter should come out. Yeah? Synaptobrevin. Thank you. Right? He is very sensitive about P. Right? So synaptobrevin binds with the syntaxins, and their binding is dependent on calcium influx, and of course calcium influx is dependent on approaching depolarizing waves. Is that right? Now let's sum it up what we have discussed. We have just discussed that when action potentials are produced as electrical stimulus on the motor neurons, wave of depolarization rapidly approaches the nerve ending. When nerve ending is depolarized, the depolarization sensitive calcium channels are activated, a lot of calcium influx occur. This calcium influx stimulates the proteins on vesicular membrane and nerve ending membrane, help them to interact with each other, so that vesicular membrane and nerve ending membrane, they fuse with each other and they produce a hole through which what will happen? Neurotransmitter will come out. In this way, action potential produces calcium influx, and calcium influx produces neurotransmitter release. This is simply 3 steps. Approaching action potential produces calcium influx in the nerve ending, and that calcium influx produces release of neurotransmitter. So these are all presynaptic events at neuromuscular junction. Now, so what really happens, I will draw multiple areas here, that so many vesicles will fuse there, and from these vesicles what is being released? Yes please, acetylcholine. Is that right? Now acetylcholine has 2 parts. One part is suppose choline, this part is choline, and this is acetate. This is choline and this is acetate, and of course this choline and acetate, they must have choline also. Am I clear? Okay. Now acetylcholine is released. Now this acetylcholine concentration is more near the presynaptic membrane, and concentration here is less. So things move from high concentration to low concentration. So acetylcholine, which is released by the nerve ending, it moves from the presynaptic membrane towards the postsynaptic membrane, which is basically here muscle and plate. So by simple diffusion mechanism, neurotransmitter or acetylcholine diffuses from the presynaptic area to the postsynaptic area, and this gap between the presynaptic membrane and postsynaptic membrane is called synaptic cleft. What is it called? Synaptic cleft. Is that right? Now what we'll see, what are the events which happen here. Actually, here are acetylcholine receptors. Is that right? If someone is talking to you, you must have ears. So here is the tongue and here are the ears. Unfortunately, it has only tongue and it has only ears, maybe fortunately, right? So this is one-way transmission, right? So the acetylcholine sensitive special type of receptors are here. These receptors are very, very special type of channels. One, two, three, four, 5. These are pentameric proteins. Pentameric proteins mean these are special type of 5 proteins come together and making a channel. And of course there is alpha protein. There are 2 alpha protein, beta, gamma, delta proteins. No need to remember that detail, but if you want to, it's so simple. 2 alpha plus 1 beta, 1 gamma, 1 delta. So all these chains, 2 alpha chains, 1 beta chain, 1 gamma chain, 1 delta chain, they assemble together to make this special type of channel. Now, actually on the sides of these, there's one point here and there is another point here. These are the points which act as receiving point for acetylcholine. If there is no acetylcholine binding here, the central pore remain closed. But as soon as acetylcholine diffuses here and binds here, they become loaded with acetylcholine. So then these channels will open. It means these channels are not voltage gated, they are ligand gated. Ligand means some specific chemical substance. These are acetylcholine operated ion channels. What are these? Acetylcholine sensitive ion channels or acetylcholine gated ion channels, or simply ligand gated. It's a type of ligand gated ion channels. Now what really happens? Let me make it more simple. Let's suppose I show 3 of these channels here. What are these? These channels are primarily acetylcholine receptors. What are these channels? These are acetylcholine receptors. These are expressed on nerve. These are expressed on the muscle end plate, right, at the postsynaptic junction of neuromuscular junction. Is that right? So these are the ears of the what? Muscles to listen to the chemical transmission of cholinergic chemical transmission of nerve, motor nerve ending. Right? As soon as this acetylcholine binds at these places, what happens? These channels open. When these channels open, they allow the cations to move. Right? These channels allow the movement of the cations. Now cations which are outside are sodium, and cations which are inside are potassium, because they allow the free movement of the cations, right? So sodium will move in and potassium will move out. Is that right? But before the acetylcholine could bind with these channels and before these channels could open, this muscle was having resting membrane potential of minus 70 millivolt. Let's suppose this muscle was having resting membrane potential of minus 70 or minus 80 millivolt. Now listen. This was having resting membrane potential of minus 80 millivolt. So resting membrane potential of the muscle membrane is more near to the equilibrium potential of potassium or more near to the equilibrium potential of sodium. Potassium, potassium has the equilibrium potential of minus 85 millivolt and sodium has equilibrium potential of plus 65 millivolt. So it means before the acetylcholine mediated stimulation of these channels, there was resting membrane potential on the motor end plate, and this resting membrane potential was a voltage which was more near to the potassium equilibrium potential. So as soon as acetylcholine is released and diffuses down and stimulate the acetylcholine receptors. What are acetylcholine receptors? Acetylcholine receptors are acetylcholine sensitive cationic channels. Another thing, in these pockets, if I make this channel here, if this is the channel, right? I told you there's one pocket here, another pocket here. In these pockets, not only acetylcholine can fit, but this is acetylcholine, and this part of the acetylcholine, this triangular part of acetylcholine fits into this area. There is another chemical substance which is like this. That alone can also fit here and stimulate it. This substance is not acetylcholine. Yes. This substance is nicotine. So these channels can also be stimulated by nicotine. So these are also called nicotinic channels. They are also called nicotinic acetylcholine channels because there are other channels of there are other receptors for acetylcholine which can not be stimulated by nicotine. They can be stimulated by muscarine. I will not go into that detail today, but for a while I just mentioned that these acetylcholine receptor channels which are present on the motor end plate, these channels can also be stimulated by nicotine. So they are also called nicotinic channels. And these are acetylcholine sensitive channels which also responds to nicotine. So these are called nicotinic cholinergic receptors. What are these called? Receptors. Nicotinic cholinergic receptors. So what are nicotinic cholinergic receptors? These are nicotine as well as acetylcholine sensitive receptor channels which are present on the motor end plate where acetylcholine will bind and these channels will open. These are cationic channels. They allow the cations to move across the membrane down their electrochemical gradients. We were discussing that as soon as when these channels are opening, just before they open, the resting membrane potential of the motor end plate is near the equilibrium potential of potassium and it is far away from the equilibrium potential of the sodium. You know already from previous lectures that whenever a membrane becomes highly permeable to a specific ion, that ion will move down its electrochemical gradient and try to take the membrane potential towards its own equilibrium potential. Now, because Now sodium can go in and potassium can go out. Is that right? So sodium influx will try to take the membrane potential towards its own equilibrium potential, and potassium will try to maintain the membrane potential near its own equilibrium potential. As soon as these ion channels open, membrane is already near to the potassium equilibrium potential. So potassium will not move in massive amount, but membrane is far away from sodium equilibrium potential. So sodium will move in very high concentration from outside to inside. Am I clear? Right? So even though there is a little potassium efflux, but mainly there is a heavy influx of sodium. So this sodium which is coming out through these acetylcholine receptors, the sodium influx make progressively, as more and more sodium is coming in, it progressively make the membrane potential less and less negative, and it become minus, from minus 80 it become minus 60, minus 40, minus 20, and most of the time it become 0 millivolt. This is 0, not anything else. 0 millivolt. So it means membrane which was negatively polarized, now it is depolarized. So if there is enough acetylcholine present, and there are enough acetylcholine receptors and channels responsive, then enough sodium will come in to depolarize the membrane and take the membrane potential from the resting membrane potential to, let's suppose, 0 millivolt. Am I clear? There's no problem in this. Now one thing which is very important to understand, usually when action potential come on the nerve ending, lot of calcium influx occur and thousands of the vesicles release lot of acetylcholine, and lot of acetylcholine stimulates many cholinergic postsynaptic nicotinic receptors, and lot of sodium comes in and depolarizes the membrane. Now this is not action potential. When resting membrane potential from minus 80 has gone up to 0, it is not action potential, it is just a local potential, and because this local potential is produced at the end plate, so we simply call it end plate potential. What is it? Potential. So we say when resting membrane potential from minus 80 goes to 0 millivolt, so 0 millivolt is motor end plate potential. Is that right? Or simply we call it end plate potential, motor end plate potential. Now one thing which is very important to know, this end plate potential from minus 80 to 0 is not produced by the contents of only one synaptic vesicle. Let's imagine what is the smallest fluctuation which can be produced here. Smallest fluctuation can be produced by release of the contents of single vesicle. Let's suppose, theoretically speaking, that if only one vesicle releases its content and no other vesicle release, then very small amount of acetylcholine will come. There will be very little stimulation of cholinergic receptor, very little sodium will come in, right? And in that case, membrane potential will fluctuate maybe only 0.4 millivolt. Is it a large fluctuation? Even this fluctuation is less than 1 millivolt. This fluctuation is less than 1 millivolt. Again, let me repeat it. If, hypothetically speaking, if only one vesicle releases its acetylcholine, that is such a small amount of acetylcholine that it will do so little stimulation of cholinergic receptors and channels that such a small amount of sodium influx will occur that there will be very little fluctuation in local potential, maybe only 0.4 millivolt. Right? This type of very small fluctuation by single content of the single cholinergic vesicle is called miniature end plate potential. What is it called? Miniature end plate potential. So we can say that when, now listen carefully, what is miniature end plate potential? Miniature is a very small end plate potential. This is the smallest possible. You cannot have end plate potential less than this, right? Miniature end plate potential is really a miniature end plate potential, very small end plate potential, which is produced by the release of content of acetylcholine from a single vesicle which produces so less stimulation of cholinergic receptors, so small amount of sodium influx is produced, there's very little fluctuation in resting membrane potential, or very little membrane fluctuation is produced. For example, if it was minus 80 millivolt, it will become just minus 79.6 millivolt. It's a very small fluctuation. We call it miniature end plate potential. Now what really happens? This is also called quantal release. This is the smallest quanta of acetylcholine which could be released. You cannot release less than that. At least one packet of acetylcholine should be released. So when one vesicle or one packet of acetylcholine or one quantum of acetylcholine is released, this is responsible to produce miniature end plate potential. But in fact, in a healthy neuromuscular transmission, the neuronal depolarization produces so much influx of the calcium in the nerve ending that thousands and thousands of many of the vesicles are released, and many miniature end plate potentials are produced simultaneously, and they are summated together. They are added together, right, to produce the full end plate potential. But again, you have to remember, this full end plate potential is just a local fluctuation in the end plate region. Full end plate potential is not action potential, because it is not due to voltage gated sodium channel. It is produced due to ligand gated sodium channels. But once enough sodium has come in, what this sodium will do? This sodium will go to the surrounding area, is that right? And on the neighboring area, there are, what are these? Voltage gated sodium channels. These are present on the flanks of motor end plate, right? So what really happens, that when motor end plate undergoes the process of depolarization, and this depolarization is end plate potential. This end plate potential actually takes the flanking part of the membrane on the sides of the motor end plate. It takes that part of the membrane from resting voltage to threshold voltage. Is that right? So what really happened, that I can write here that end plate potential here and end plate potential here, it will take the resting membrane potential to threshold potential. This end plate potential will take on the flanking region resting membrane potential up to, yes please, threshold potential. At threshold potential, right, what will happen? Voltage gated sodium channels will open and heavy amount of sodium influx will start. And as you know from previous lectures, that when sodium influx occurs, heavy sodium influx occurs, due to the voltage activation of the voltage gated channel on the muscle membrane, right, this sodium will stimulate the next area. Is that right? This depolarizing current will take the neighboring part of the muscle membrane to threshold, and this will undergo depolarization, and this will take the next area to threshold, and that will undergo depolarization. So what really happens, that wave of depolarization will rapidly sweep over all the muscle cell membrane. Is that right? How that depolarization triggers the muscle contraction, that we will study in future lectures when I will teach you electromechanical coupling, that how the electrical activity eventually gets coupled with mechanical contraction. Is that right? Not today. Today our discussion is only how the information is transferred from the motor neurons to the muscle membrane, right, which is called neuromuscular transmission at neuromuscular junctions. Is that right? There's no difficulty. So this was the basic concept of neuromuscular junction. Let me revise it for you rapidly. What really happens at neuromuscular junction? When in spinal cord, cell bodies or neurons, motor neurons are stimulated, these motor neurons generate action potentials, and these action potentials are going down the membrane of the motor neurons, right? So when these action potentials, wave of depolarization approaches the, what is this? Nerve terminal. There is depolarization sensitive calcium channels present only at the nerve ending. These depolarization sensitive calcium channels were not present throughout. They were only present at the nerve ending. So nerve ending develops calcium influx. Then calcium influx mediated stimulation of vesicular proteins and nerve ending membrane proteins occur, and these proteins interaction produces the fusion of vesicular membranes with the nerve ending membrane, and neurotransmitters are released. As soon as neurotransmitter is released, here the neurotransmitter was acetylcholine. Acetylcholine is released, it diffuses down the synaptic cleft to the motor end plate. There at the motor end plate, it finds nicotinic cholinergic receptors, which are special ion channels which are sensitive and operated by the acetylcholine. As soon as acetylcholine bind with these channels, this allows the movement of cations, right? Potassium will move out and sodium will move inside the cell. But because membrane was initially more near to the potassium equilibrium potential and far away from the sodium equilibrium potential, so heavy sodium influx is generated, and that takes the resting membrane potential of the end plate to not here is not threshold, it takes to depolarization. Is that right? Remember, if there's only one vesicle releases its acetylcholine, right, there is very small influx of sodium produced, and very small fluctuation in the local potential is called miniature end plate potential. But normally many vesicles of acetylcholine release their contents, and a lot of cholinergic nicotinic receptors are activated, a lot of sodium influx occur, and resting membrane potential fluctuate from -80 value to approximately 0 millivolt. So its negative polarity is completely lost. So membrane in this region, motor end plate region, will become completely depolarized. Once this membrane becomes depolarized, this is called end plate potential, local potential. It is still not called action potential. Then this motor end plate potential is responsible to take the neighboring resting membrane potential to threshold, and neighboring membrane, the muscle cell membrane, which is in the neighboring region of end plate area, this neighboring membrane is having lot of voltage sensitive sodium channels. So when end plate potential take the resting membrane potential in the flanking area from towards the threshold, and as soon as threshold potential is achieved, voltage gated sodium channels open and heavy influx of sodium is produced, and that depolarizes that part of the membrane, and that depolarizes the next area, and so forth. Wave of depolarization moves on the muscle membrane, followed by, of course, wave of repolarization. Right? We have discussed it already how it happens. So in this way, actually the action potentials which were coming on the neuronal membrane, right? Those action potential were electrical signals. By release of the neurotransmitter, those signals were converted into chemical signals, and chemical signals approach the postsynaptic membrane and again produce electrical signals, and when these electrical signals start flowing with, say, action potential of the neuron through the neuromuscular transmission, have produced action potential of the muscle membrane. This is how neurons talk intimately to the muscles. Is that right? But there's one thing. This talk should have its limit, isn't it? It should not be continuous talk. So, actually, this acetylcholine which has been released, it should not stay there forever. Alright? For this purpose, nature has been very kind to us, that there are special type of enzymes, and these enzymes are present on the membrane of, as well as in the extracellular matrix of neuromuscular junction. In the neuromuscular junction, these special type of enzymes are present. Where they are present? They are present within the extracellular matrix, as well as they are present on motor end plate membrane. These enzymes, what they will do, that they will catch the acetylcholine and break it down into choline and acetate. They will break it down into choline and acetate. Acetate will diffuse away through the blood. It may go to the liver and used for some other purposes. This choline is very variable. It will be again pumped back. Is that right? So these are the enzymes which are responsible to catabolize the neurotransmitter, and acetylcholine is broken by the action of these enzymes into choline and acetate. Acetate diffuses out, and choline is again reabsorbed. Why? Because this nerve ending cannot produce the choline, but it can produce acetate from its mitochondria. It's so intelligent. It can produce its own acetate, so it doesn't really uses energy to pick it up. But because it cannot synthesize choline, so 50% of the choline, acetylcholine, which is catabolized, its choline is reuptaken, so that it can be recycled, and again resynthesis of acetylcholine can be done. These enzymes are called, yes, acetylcholinesterases. What are the name of these enzymes? These enzymes are called acetylcholinesterases. These are acetylcholinesterases. Right? These enzymes are present at the neuromuscular junction, and they are responsible to destroy the acetylcholine once it has done its action. So we call them simply acetylcholinesterases. Acetylcholinesterases, right? They produce the hydrolysis of acetylcholine and breakdown of acetylcholine into acetate and choline. In this way, they help the neuromuscular junction to terminate the action of acetylcholine. If neuron want more action in the muscle, then neuron will bring more action potential and release more acetylcholine, and again action potential will be set up. Am I clear? There's no problem here. Right, let's have a break. Thank you.